Friends, again welcome to my channel. Today we are going to discuss about one of the important topic from the immunology, that is the structure and function of MHC class first molecules. Now, in this MHC class first molecules, these are present as an surface molecules, or on the surface of the antigen presenting cells there is the presence of this MHC molecule. These are the cell surface glycoproteins. Majorly these major histocompatibility complex, these are present on the cluster of gene rearranged within the long continuous stretch of DNA on the chromosome number 6. Means the genes responsible for the synthesis of these MHC molecules, which are the surface glycoproteins, these are located on the chromosome number 6 in the human beings. And preferentially these are present on the surface of antigen presenting cells for the purpose of representation of antigens towards the T lymphocytic cells. Means these are essential for the antigen presentation and processing. And majorly these MHC molecules, these are also responsible to differentiate in between the individuals, because these are characterize the individual cells. The MHC cluster of genes for the MHC molecule which is present within the human beings, it is known as the human leukocyte antigen complex, that is the HLA complex. And preferentially these MHC molecules, these are get categorized into the three different types, that is the MHC class first, class second and the class third molecules. Majorly the genes which are responsible for expressing these MHC molecules, these are having the variations, or as we say that these are responsible to differentiate in between the individuals. For this variability, preferentially there is the presence of the multiple alleles for these genes. There is presence of multiple alleles, that is these genes, these are polygenic and polymorphic in nature, means there is the observation of polymorphism within these genes, that is presence of the large number of alleles for each of these genes. These are present on the chromosome number 6. A large number of alleles for these genes, these are present in the population for each of the gene, and each individual, these are inherits a restricted set of alleles for his or her parent. Means there is the specificity of this. This set of MHC genes, they tend to be inherited as a block or as an haplotype. And majorly there are relatively infrequent crossover events at this locus, means there is no any type of the crossover events, these are occur within that in that region. Now we are switch over to the detailed structure of this HLA complex. Now majorly the gene locus for this major histocompatibility complex which is present within the human beings, which is known as this HLA complex or the human leukocyte antigen complex, it is located on the short arm of the chromosome number 6, and there is the presence of the genes that are critical to the immune function, means these are participated within the immune function, that is majorly these are responsible for the antigen presentation towards the immune cells. And as majorly these are classified as the class first molecule. The gene locus for this class first molecule includes the HLA-A, HLA-B and the HLA-C. Means these three gene locus A, B and C, these are responsible to give the class first MHC molecule. In case of the class second, the gene loci which are present, it is the HLA-DR, HLA-DQ and the HLA-DP. That is the DR, DQ and DP, these gene loci, these are responsible to give the class second MHC molecule. All of these are present within a HLA-D region of the HLA complex. The third type of MHC molecule, that is the class third MHC molecule, majorly these are responsible for encoding the different types of the complement factors which are the part of the complement system, that is the tumor necrosis factor, then the complement proteins, that is the C2, C4, all of these. The class first MHC genes, these are responsible to encode the glycoproteins, and preferentially these glycoproteins, these are observed on the surface of all of the nucleated cells. Means within our body different nucleated cells which are present, these are responsible to representing this class first MHC molecule, and majorly these class first MHC molecules, these are responsible to represent the endogenous antigens towards the T cytotoxic, that is the CD8 positive T lymphocytic cells. Means in other words we can say that these class first MHC molecules, these are essential for the antigen presentation and the processing of the endogenous antigen. Presentation and processing of endogenous antigen. In reverse to that, whenever we are considering about the class second MHC molecule, these are responsible to encode the glycoproteins which are predominantly present on the surface of the antigen presenting cells, like that of the dendritic cells, B lymphocytic cells, macrophage cells, which are the professional antigen presenting cell and responsible to represent the antigens towards the T lymphocytic cells, and these are processing and present the exogenous antigen, that is those antigens which are entered within our body from the outside in the form of bacterial cell or the toxins. Such type of the exogenous antigens, these are processed and presented by these MHC class second molecules towards the CD4 positive T helper cells. Now these class third MHC molecules, these are responsible to encode the several different complement protein. Now we are focusing on the class first MHC molecules. Whenever we are discuss about the structure of class first MHC molecule, it mainly consists of the two polypeptide chains, like that of which are present within the T lymphocytic cells or the T cell receptor of the T lymphocytes, that is the alpha and beta. In the same way here, this class first MHC molecules, these are contains the two different polypeptide chain, that is alpha and beta, means it is an heterodimer. It is an heterodimer. These two chains, that is the alpha and beta chains, these are connected together by the non-covalent bond. This alpha chain, it is characterized as an internal membrane glycoprotein, and the molecular weight of this alpha chain, it is about the 45,000 Dalton within the human beings. And the beta chain on the other hand, it is an extracellular microglobulin. It is an extracellular microglobulin with a molecular mass of 12 kilodalton. Means the largest polypeptide chain which is present, it is the alpha chain. Only the alpha chain it is present, and the another molecule, that is the beta microglobulin, it is located on the another gene loci, or it is only the non-covalently attached with the alpha chain. Now within the alpha chains, there is the presence of the three different domains. Majorly by the folding of the polypeptide chains, there is the formation of this domain structure, that is the alpha 1, alpha 2 and the alpha 3 domain. Each of these domains contains roughly about the 90 amino acids. The N terminal, that is the amino terminal end of the alpha chain, it is the place of alpha domain, and that is the alpha domain first. It is the place of alpha domain first, and the alpha 2 and the alpha 3, these are present after the alpha 1. The alpha 2 domain, it is characterized by the formation of a loop of 63 amino acids, and the loop it is formed due to the intra-chain disulfide bond. Means this domain structure it is get resulted due to the intra-chain disulfide linkages. The alpha 3 it also contains the disulfide bonds enclosing about 86 amino acid, and the alpha 1 and the alpha 2 domains interact to form a peptide binding cleft, that is the binding site for the antigen. That is, whenever these are responsible to process and present the antigens towards the T lymphocytic cells, these processed peptides, these are bind within that region, that is which is made up of the alpha 1 and alpha 2 region, which is called as the peptide binding cleft. as we know that this class first MHC molecule, these are responsible to represent the endogenous antigens towards the T cytotoxic cells. The alpha 1 and alpha 2 domains interacts to form this deep groove on the top, which is the peptide binding cleft, and it can bind the antigens having, or the that is the peptide antigen having, which are the 8 to 10 amino acid in length, means the shorter peptides. These are going to represent by the MHC class first molecules. Again, this alpha chain, it also consists of a stretch of 26 hydrophobic amino acids, which is responsible to hold this alpha chain on the plasma membrane. Means as these are the 26 hydrophobic amino acid, it is nothing but the transmembrane region, because these MHC molecules, these are present on the surface of antigen presenting cells, or in other words we can say that it is present within the membrane bound form. This transmembrane segment of 26 hydrophobic amino acid is represent as a form of helix at the hydrophobic region of the plasma membrane. An intracellular domain of the carboxy terminal of alpha chain, it is located inside the cell, that is the cytoplasmic tails. The part which is present inside the cell, it is considered to be the carboxy terminal domain. Inside the cell, it contains about 30 to 40 amino acid, or it is made up of 30 to 40 amino acid. And majorly these amino acids, these are hydrophilic in nature. The amino acid present within the cytoplasmic part of the cytoplasmic tails, these are hydrophilic in nature. Again, within the human beings, within this HLA complex, the beta chain, it is non-polymorphic. It is non-polymorphic, means the polymorphism, it is not present within this beta chain or the beta 2 microglobulin, which is the part of the MHC class first molecule, and it is dimorphic in mice. The alpha 3 and the beta 2 microglobulin are structurally similar to the immunoglobulin C domain and also characterized as a disulfide loop. Majorly these are having the similar domain structure to that of the immunoglobulin molecules. And there is the non-covalent interaction in between the alpha 3 domain of the alpha chain and the beta 2 microglobulin. Here it is an structure of the MHC class first molecules. Within this you can observe there is the transmembrane segment, which is made up of the hydrophobic amino acids, that is the 26 hydrophobic amino acid. These are responsible to synthesize this transmembrane segment. The cytoplasmic tails, which is consist of the 30 to 40 hydrophilic amino acid. Then the alpha chain, which is the main polypeptide chain, which is present within this class first MHC molecule, it is get folded to give the domain structure. This domain structure, it is similar to that of the immunoglobulin domains, and there is the formation of 3 loop structure or the domain structure, which is characterized as the alpha 1, alpha 2 and the alpha 3, and all of this domain structure, it is get resulted due to the disulfide linkages. And the alpha 2 and the alpha 1 domain of the alpha chain, it is responsible to give the peptide binding cleft, within which the peptides having the length 8 to 10 amino acid, they can fit, and these are get represented towards the T cytotoxic or the CD8 positive cells, that is preferentially the endogenous antigens. And the beta 2 microglobulin, which is the another component within the MHC class first molecule, it is non-covalently attached with the alpha 3 domain. This beta 2 microglobulin is a protein encoded by the highly conserved gene located on the different chromosome. Means these are not located on the specifically within the this class first, or these are not located on the this region, that is the HLA A, B and C region, but it is located on the another gene. It is a type of protein. Means here we can say that majorly within the MHC class first molecule there is a single polypeptide chain, it is present, and this single polypeptide chain, it is associated with the another protein which is encoded by the another gene located on the different chromosome. The beta 2 microglobulin, it is similar in size and organization of the alpha 3 domain, and it is the reason why it is get non-covalently attached with the alpha 3 domain. The beta 2 microglobulin does not contain the transmembrane region, and it is non-covalently linked with the alpha chain. Means there is absence of the transmembrane region within the beta 2 microglobulin. Now we are switch over to the functions of MHC class first molecule. Now the major function of this MHC class first molecule, it can bind with the peptide antigens, that is preferentially the endogenous antigens. And these are responsible to process this endogenous antigen and represent this endogenous antigens towards the T cytotoxins, that is the CD8 positive cells. These CD8 positive T cells, these are specific for the MHC class first molecule, and these are representing this endogenous antigen. So it is the information about the structure and the function of MHC class first molecules. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.